Okay, let's continue our discussion on the Sanger dideoxynucleotide sequencing. So, there are four test tubes. Each test tube has the in vitro DNA replication condition, and these are all the same condition tubes, right? Each tube has the template DNA, DNA polymerase, primer, and DNA nucleotide. So those alone will be enough for the replication to take place in vitro, right? So on top of that, you will add dideoxynucleotide, that strange nucleotide that's going to be incorporated, but whenever it's incorporated, it's going to stop the replication reaction, right? You're going to add that to all these four tubes. So one dideoxynucleotide per each tube. So for example, this dideoxynucleotide A will be added to this first tube. Dideoxynucleotide C will be added to the second tube. Dideoxy T to the third. Dideoxy G to the fourth tube. So we're going to add those dideoxynucleotide to all four tubes and see what happens. Okay. So each tube is labeled with whatever the dideoxynucleotide that was added to that tube, right? And you're going to take a look at the inside of the test tube and see what happens. Okay, so let's take a look at that tube and see what happens there. Okay, you're going to blow it up and take a look at it. There you go. So the template DNA is there. That's being denatured because you are heating it up, right? And once it's template DNA denatured, it becomes single-stranded. Then what's going to happen? The primer can find the matching sequence, and then that's the primer binding, right? That's going to take place. So you're going to cool down the temperature, and then the primer can come and bind, right? So temperature is going down, primer binding happens, right? And then, so the DNA polymerase, it's grabbing the tail of the primer, and then it's going to start the replication. That's good. Yeah, it goes fine, but until the dideoxy A nucleotide was added. Okay, so it added these nucleotides. They were normal DNA nucleotide, but whenever the dideoxynucleotide is added, your DNA replication is terminated. So the first replicated product is only this long. Okay, but is that the end? No, there could be multiple DNA replications that can happen. See? Okay, so you're gonna, yeah. So because there may be multiple DNA template DNA there with multiple primers and multiple DNA polymerases, so they're going to do the replication. Okay, and then they're going to have the random termination because it's not quite sure where the dideoxy A nucleotide will be added. Okay, so whenever that dideoxynucleotide A was added, it's going to be terminated, but you don't know that insertion site. So it may be inserted there, there, there. Depends on the location of the dideoxynucleotide. The termination of the DNA replication would be different, and then because of that, the size of the replicated DNA product will vary. You see that? So there are multiple DNA replication product. They will have different sizes, but the common thing is they have all dideoxynucleotide at the end of their tail, because that will terminate the replication product. Okay, that's what you expect to see from this tube. Okay, and how about the rest of the tube? Probably the similar thing happened, right? This one, since it had dideoxy C nucleotide added, so there would have been a termination wherever that was incorporated. Okay, so there will be multiple replicated product with different sizes, but they will all have dideoxy C at the end. Same goes with these four tubes as well. Now you're going to separate out the replicated product on electrophoresis gel. So you have the polyacrylamide gel there. You're going to add a sample, this sample to here, next sample to there, third sample here, fourth sample there. So you're going to sort them out based on the sizes. That's what electrophoresis will do, right? So there you go. Electrophoresis is done. Okay, loading dye has migrated from negative to positive side. Now you're going to visualize them. Okay, so the band will show up because there will be some kind of a signal generating probe that's added there. Okay, the probe can be added to the primer, or probe can be added to the nucleotide. So anyway, you can visualize the band. So what do you see there? So out of so many replicated products, okay, you can see this one is the smallest because that migrated furthest distance, right? And then that is the largest. Okay, doesn't matter which tube they came from. Okay, if you sort out all these fragments, this is the smallest, and that's the largest, right? Okay. Another information you can get out of this fragment is what nucleotide they ended with, right? So this guy ended with dideoxy T. Okay, that one as well. So these guys all ended with dideoxy-T. These guys ended with dideoxy-A, right? And these guys ended with dideoxy-C. These guys ended with dideoxy-G. So if you can read what nucleotide they ended at the end from the smallest to the largest, then that's going to be the order of the nucleotide sequence. How are you going to do that? Look at it. So yeah, you read it from the bottom. Next one, okay? And then you read what nucleotide they ended with. Okay, that's what is being done here. Okay? So what is done is, so this one had dideoxy-T. So the smallest fragment had a T nucleotide at the end. Next smallest one was this one that had a G, okay, G at the end. Next one was this one, T again, okay. And the next one was this one that had A there. So you're just reading it continuously, like T, G, T, A, okay. And then next one was what? G again, G, and the next one was what? A again. So you just continuously read from the smallest to the largest with what nucleotide they ended their replication with. So that's the sequence of this. You just wrote on, okay? And why is that? That's the sequence of the newly made strand, newly replicated strand, because they told you what nucleotide they finished their replication with. Okay, so actually that is a sequence of the newly replicated strand. Okay, so that's how that sequencing works. So you can do that typically about 200 to 500 base pair or nucleotide long within a given sequencing analysis. Okay, that's how it works. Does it make sense? And there's another animation there. We're going to play the animation in the next lecture because this is a little modified single dideoxy sequencing, so which is more efficient. So we're going to play that later on. But you're welcome to play that if you want to right now. So going back to so that's the main function of this dideoxynucleotide because it terminates the replication process, and the termination is really important because through that termination you're going to generate all these nucleotides with different sizes, and different sizes come from where that dideoxynucleotide was incorporated. But as soon as it's incorporated, your replication is terminated. Once you have these replicated products made, then what? You just sort them out through the electrophoresis from the smallest to largest, right? But the thing is, you don't have just a single tube. You have 3 more tubes with different dideoxynucleotide, right? So you're going to sort out all those replicated products from 4 different tubes together. Okay, so that's how the sequencing will work. Okay, so basically it's this, right? Replication tube. There were 4 of them. Okay, so they all have the same replication condition except for the fact that they have different dideoxynucleotide added, right? And they have different replication product with different dideoxynucleotide incorporated at the end. Okay, you're going to sort them out on the gel, and then from the smallest to the largest, and then with what dideoxynucleotide added at their tail, you just call them from like a C, G, AA, G, T, C, A. So you can write out that sequence. That's how that sequencing analysis works. Okay, so that's the end of the lecture 9. But we're going to continue more discussion about the sequencing method of different kinds in the lecture 10. All right, thanks.